As our beloved cats grow older, they can develop changes in their brains that mirror those seen in humans with Alzheimer's disease. This fascinating connection has led scientists to study the brains of elderly cats, hoping to unlock secrets about both feline and human aging. In this video, we'll explore the latest research on neuropathology in aging cats, what it reveals about cognitive decline, and how these findings could help us better understand Alzheimer's disease in people. Let's embark on a journey through the aging feline brain and discover the surprising similarities it shares with our own. Cognitive Dysfunction Syndrome, or CDS, is the term used to describe dementia-like symptoms in aging cats. Just as Alzheimer's disease affects memory and behavior in humans, CDS leads to changes in a cat's behavior, such as confusion, altered sleep patterns, and even house soiling. These symptoms are more than just signs of old age. They reflect real changes happening inside the brain. Understanding CDS in cats not only helps veterinarians and pet owners provide better care, but also offers a unique window into the processes underlying dementia in humans. Alzheimer's disease is the most common cause of dementia in humans, marked by memory loss, confusion, and changes in thinking. In the brain, Alzheimer's is characterized by two main features: the buildup of beta-amyloid plaques outside neurons and the formation of neurofibrillary tangles made of hyperphosphorylated tau protein inside neurons. These changes disrupt communication between brain cells and eventually lead to cell death, causing the symptoms we recognize as dementia. Scientists have long sought animal models to study these processes, and now cats are coming into the spotlight. Cats are increasingly recognized as valuable models for studying age-related brain changes. Unlike laboratory rodents, cats naturally develop both beta-amyloid and tau pathologies as they age, much like humans. This makes them especially useful for understanding the progression of dementia and testing new treatments. By examining the brains of cats with and without cognitive dysfunction, researchers can draw important parallels to human Alzheimer's disease, potentially accelerating discoveries that benefit both species. In a recent study, scientists collected and examined the brains of 55 cats of various ages, some with confirmed cognitive dysfunction syndrome. Using advanced techniques like immunohistochemistry, they looked for the presence and distribution of beta-amyloid and hyperphosphorylated tau proteins in different brain regions. Their goal was to see how these proteins accumulate with age and whether their patterns resemble those seen in human Alzheimer's disease. The brains used in this study were collected post-mortem from cats that had been euthanized for medical or behavioral reasons, with full consent from owners or shelter managers. Ethical approval was obtained from the relevant veterinary committee, ensuring that all procedures met high standards for animal welfare. This careful approach allows researchers to study naturally occurring disease without causing additional harm to animals. Researchers used a technique called immunohistochemistry to detect specific proteins in thin slices of cat brain tissue. By applying antibodies that bind to beta-amyloid and tau, they could visualize where these proteins were located and how much had accumulated. This method allows for precise mapping of pathological changes across different brain regions, providing a detailed picture of how aging affects the feline brain. Beta-amyloid is a protein fragment that, when it accumulates outside neurons, forms plaques, a key feature of Alzheimer's disease. In cats, researchers found both intracytoplasmic inside cells and extracellular outside cells deposits of beta-amyloid. Interestingly, elderly cats had more and larger extracellular deposits, especially in the cortex, while younger cats had more intracytoplasmic deposits. This pattern closely mirrors what is seen in the early stages of human Alzheimer's. While humans with Alzheimer's often develop dense, core plaques of beta-amyloid, cats typically show a more diffuse pattern of extracellular deposits. These diffuse plaques are thought to represent an earlier stage of the disease process. In rare cases, older cats did develop more compact, plaque-like structures, but most showed the diffuse type, especially in the cortex. This suggests that cats may model the early progression of amyloid pathology seen in humans. In both cats and humans, Amyloid deposits begin in the cortex and later appear in the hippocampus, a region crucial for memory. The cerebellum, responsible for coordination, is affected only in the latest stages. This progression mirrors what is seen in human Alzheimer's, supporting the idea that cats can serve as a natural model for studying the disease's early development and spread within the brain. Younger cats showed more intracytoplasmic beta-amyloid deposits, especially in cortical neurons. As cats age and extracellular amyloid builds up, the amount of intracytoplasmic amyloid tends to decrease in these regions. This shift from intracellular to extracellular deposits is also observed in human Alzheimer's, suggesting a similar disease process. In some brain regions, like the hippocampus and cerebellum, intracytoplasmic amyloid actually increased with age. Tau is a protein that helps stabilize the internal skeleton of neurons. In Alzheimer's disease, tau becomes abnormally hyperphosphorylated, causing it to form tangles inside cells. In cats, researchers found both normal and hyperphosphorylated tau in various brain regions. The presence of hyperphosphorylated tau, especially in the form of pre-tangles, was more common in older cats and those with cognitive dysfunction syndrome. Pre-tangles are early forms of tau pathology, appearing as intracytoplasmic deposits in neurons. In this study, pre-tangles were most frequently found in the cortex of elderly cats, especially those diagnosed with cognitive dysfunction syndrome. The number of pre-tangles increased with age and was significantly higher in cats with CDS compared to age-matched controls. This strong association mirrors findings in human Alzheimer's, where tau pathology is closely linked to cognitive impairment. For the first time, researchers identified tau protein inside the nuclei of cat neurons, both in phosphorylated and non-phosphorylated forms. Intranuclear tau was most abundant in younger cats and decreased with age, especially in those with pre-tangles. This is a fascinating finding, as nuclear tau is thought to play a protective role in DNA maintenance, and its loss may contribute to neurodegeneration. To ensure their observations were accurate, researchers used several controls. They performed isotype controls to rule out nonspecific antibody binding and used dephosphorylation treatments to confirm that nuclear tau labeling was truly due to phosphorylation. Western blot analysis further validated the presence of hyperphosphorylated tau in nuclear and cytoplasmic fractions. These rigorous methods strengthen the reliability of the study's conclusions. The study used advanced statistical methods to analyze the data, including ANOVA, t-tests, and chi-square tests. These analyses revealed significant age-related differences in amyloid and tau pathology, as well as strong associations between pre-tangles and cognitive dysfunction. By quantifying protein loads and burdens in different brain regions, the researchers could draw meaningful conclusions about the progression of disease in cats. The patterns of amyloid and tau accumulation in cats closely resemble those seen in human Alzheimer's disease. Both species show early amyloid buildup in the cortex, followed by spread to the hippocampus, and tau pathology that correlates with cognitive decline. However, some differences exist, such as the onset location of tau pathology. These similarities and differences make cats a valuable, but not perfect, model for studying dementia. Like all research, this study had limitations. The brains were only examined post-mortem, providing a snapshot rather than a timeline of disease progression. Some cats lacked detailed medical histories, and variations in brain sectioning made it difficult to analyze certain regions in detail. Despite these challenges, the study remains the largest of its kind and provides valuable insights into feline and human brain aging. The discovery that cats naturally develop both amyloid and tau pathologies opens new avenues for Alzheimer's research. By studying these processes in cats, scientists can test hypotheses and potential treatments in a model that closely mimics human disease. This could lead to earlier detection, better therapies, and improved quality of life for both cats and people affected by dementia. Understanding the brain changes that occur in aging cats can help veterinarians and pet owners recognize and manage cognitive dysfunction. Early intervention, environmental enrichment, and supportive care can make a significant difference in the quality of life for senior cats. As research continues, new strategies may emerge to slow or prevent cognitive decline in our feline companions. The study of aging in cats not only deepens our understanding of feline health, but also sheds light on the mysteries of human Alzheimer's disease. As we care for our aging pets, we are reminded of the shared biological pathways that connect all mammals. By continuing to explore these connections, we move closer to unraveling the complexities of dementia and improving the lives of both cats and humans.